The last group of organelles we're going to talk about are those that are involved in energy processing. So that includes the mitochondria and chloroplast. In this unit, we're just going to talk about their structure and define what they do. And then in the next unit, we're going to talk in detail about how they work. Okay. So the first one we're going to talk about is the mitochondria. Mitochondria, I already had you write this down. Write it down again. Mitochondria are found in both plants and animals. Mitochondria are found in all eukaryotes. And the function of mitochondria is to do cellular respiration. Cellular respiration is the process that takes the energy from our food and releases it and passes it to the molecule ATP, adenosine triphosphate. That's that RNA nucleotide that's used for energy exchange in our cells. So mitochondria process sugar and oxygen. They make water and carbon dioxide as waste products and release the energy from our food so our cells can use it. And all eukaryotes have to do this, including plants. Okay. So the function of the mitochondria is to do cellular respiration. You also need to know the structure of the mitochondria. Mitochondria actually have two layers of membrane. They have an outer membrane, they have an inner membrane, and then there's actually a gap in between these. It's called the intermembrane space. And then the very inside is called the matrix. So even though mitochondria have membranes, they're not part of the endomembrane system. They don't make little bubbles. They don't pinch off vesicles. They do not receive vesicles from the endomembrane system. But what they do have is they actually have their own DNA and they have their own ribosomes. Now, mitochondria can't exist without any genes from the nucleus. They need some proteins that are made from genes in the nucleus, but they can make some of their own proteins themselves. Other organelle involved in energy processing are the chloroplasts. Now, chloroplasts are found in plants and photosynthetic protists, but are not found in animals. The function of the chloroplast is to do photosynthesis, to take energy from the sun and transfer it to energy stored in organic molecules like sugar. And the pigment that does this is called chlorophyll. So chlorophyll is the green pigment that helps do photosynthesis, to transfer energy from sunlight to energy that's in sugar. Photosynthesis is actually the reverse reaction of cellular respiration. It's the exact same chemical reaction in reverse for the most part. You start with water and carbon dioxide, and using energy from the sun makes sugar molecules, and then oxygen is released as a waste product. Within the chloroplast, they have a structure that I also want you guys to know. Chloroplasts actually have a double membrane, but nothing too important happens between these two outer membranes. It's the very inside membranes called the thylakoid membranes that contain the pigment chlorophyll. And the first half of photosynthesis happens here within the thylakoid membranes. And then the second step of photosynthesis, called the dark reactions, happen in the fluid-filled space inside the double membrane but outside of the thylakoids, and this is called the stroma. Within the stroma, chloroplasts also have their own DNA and their own ribosomes and can make some of their own proteins as well. So chloroplasts are one kind of plastid. Plastids are plant organelles that contain pigments. And so in our cell lab, you'll be able to observe chloroplasts, which contain chlorophyll, leucoplasts, which are also called amyloplasts, that are the starch storage organelles in potatoes, and chromoplasts that store pigments that make red potatoes red. So you might have noticed there are several structural similarities between mitochondria and chloroplasts. They both have multiple membrane layers. They both have their own DNA and their own ribosomes. In addition to that, both chloroplasts and mitochondria can replicate within the cell, independent of the cell cycle. So a cell can grow more mitochondria or grow more chloroplasts. And all of these characteristics led people to believe that maybe these guys were actually descended from bacteria, from prokaryotes. And that's called endosymbiotin theory. So endosymbiotin theory says that originally there was a eukaryote that had a nucleus in the endomembrane system, and it was just kind of scraping along doing this very inefficient process of getting energy from food called glycolysis. And at some point in evolutionary history, instead of just gobbling up little bacteria as food, this eukaryote endosymbiosed brought this little bacteria that was really good at doing cellular respiration inside of it. And the bacteria benefited because it was protected from being eaten by other cells. And the eukaryote benefited because it was getting extra ATP produced by these little bacteria until they became one organism. And then later in evolutionary history, some branch endosymbiosed a little prokaryote that was really good at doing photosynthesis. And again, instead of gobbling it up and using all the sugar that was in it, the eukaryote allowed it to live inside of it. So the little prokaryote was protected and the eukaryote got the benefit of the sugar that was produced by the chloroplast. So these eukaryotes that had endosymbiosed mitochondria, or the prokaryotes that became mitochondria, were so much more successful than eukaryotes that didn't have mitochondria that these guys all went extinct. They couldn't compete. All eukaryotes today have mitochondria. And then plants and photosynthetic protists descended from this endosymbiosis event that brought little prokaryotes that were good at doing photosynthesis inside a eukaryotic cell. So this is a term you guys should know for the exam. Endosymbiotin theory says that bacteria that were really good at doing cellular respiration were brought inside a eukaryote that became the mitochondria. And then later, prokaryotes that were really good at doing photosynthesis got brought inside eukaryotes, and that gave rise to plants and photosynthetic protists. So we have just one more membrane-enclosed organelle that I want to talk about, and those are the peroxisomes. Peroxisomes have a single membrane. They break down and synthesize hydrogen peroxide for fat metabolism. They are kind of oddballs in the organelles because they can actually replicate independent of the cell cycle, but they don't have any DNA. They only have a single membrane. And so there's a question about whether they're just really, really ancient endosymbiotes or if they're just some organelle that can replicate independent of the cell cycle. So for the peroxisome, their function is to make and break down hydrogen peroxide and break down fatty acids. So you should be familiar with all the different organelles that can be found in eukaryotic cells, as well as knowing the difference between prokaryotes versus eukaryotes and animals versus plants. One activity that might help you practice these concepts is to kind of try to set up a symbolic cell. You could say a cell is like an office building, the plasma membrane is the walls, and the nucleus is the boss, and the ribosomes are the workers. Some analogy like that that helps you remember the function of the different organelles. The other thing that you guys should be able to think about is if there's some drug or toxin that stops a particular organelle from functioning, what effect is that going to have on the cell? What will the cell still be able to do and what will the cell not be able to do without the function of each of these organelles?